my laser pointer. Oh, it's over there. So I just, some people just tell me that when you get to the query system and it seems the Qt didn't work, and if you change to another SSH client, it might work. Okay? So, but the Qt are secure shell client. Secure shell client. Yeah. That is also one. I mean, that was the one that provided by the UITS a couple of years ago, and later they changed to Qt. I don't know the reason for that, but it seems that for the query system, that older client is working. Both of them are free, by the way, so just go to download. Okay, so let's come back to talk about the states of genetics of complex disease before we have the next generation sequencing technology. We only have this for a couple of years, and so this is a history of DNA sequencing. So you can see from the very beginning, it's the 1870s. Okay, it's the discovery of DNA. I'm not going to go through this list. I'm not a historian on that. But the point here is we start to have the ability to sequence the DNA from 1950s, and initially one person can sequence one nucleotide a year. Right, and then becomes 15, and up to here, 1970s. So where is the Sanger sequencing? Okay, you see, Sanger here is about 1,500 nucleotides per year per person. Okay, and the beginning of the Human Genome Project, which is 1990, our capacity is about 200,000 nucleotides per year per person. Okay, have to sequence a year to get those. And now, for the next generation sequencing technology we have is 100 that billion? Yeah. There's so many, the number, with so many zeros, the only time we see that is our national depths right now. So you can count the numbers. But this is the capacity of what we have right now. And in our core facility, Center for Medical Genomics, you will see next time, I think the machine itself can generate about 100 gig of data per week, and with one technician, and that is very impressive. All right, so evolution of gene discovery over the past 2 decades. So initially, people start to use microsatellites. So remember the 2 to 6 base pair and repeat elements as the marker to identify genes. And of course, there's not too many microsatellites across the human genome, and the resolution is very, very poor. So you say this region is somehow related to disease, and this is a pretty big chunk, okay? And so you really don't know which part of within this region are related to that. And later, that people start to use the SNP information as a genetic marker rather than microsatellites. And you can see that this is 6,000 SNP markers across the genome, and the resolution is still very poor. And then we got to 100,000 SNPs and 1 million SNPs. So I think before we have this next generation sequencing technology, and the most dense SNP array is from both Illumina and the Affymetrix, they are close to 1 to 2 million SNP markers on these arrays. And these are so you can get really very refined resolution, and which is 0.006 centimorgan. Okay? And with the technology developed, and the analysis changed a little bit as well. So initially, that people do more linkage, family-based linkage analysis, and later working on more on the genome-wide association studies. So you can either still using the family information or not using the family information. Okay, and this is another figure just from the same, deliver the same type of information. And you can see that for the SNPs, and there's different technologies listed here, and TaqMan, and got 1 to 10 SNPs, or you can, in one batch, or you can use SNP arrays to get to close to 1 million SNPs. And this part will be used for the candidate gene or follow-up with GWAS studies. And this part is more on the genome-wide association studies. Okay? So technologies really kind of determine the way we look at the data and the way that we find the disease-related genes. In genome-wide association studies, this is something that's a very popular a couple of years ago and still right now. And so what genome-wide association studies are commonly used to link genetic variations, mostly SNPs, with disease or traits. Okay? If the genetic variation are more frequent in person with disease, the variation are said to be associated with the disease. Okay. It's very straightforward. So in this figure, so you can see that this is a SNP. There's 2 SNPs here, okay? And I apologize for the color, although I didn't make it. It's just happens to be this way. And you can see that there's a nucleotide here, and either ACGT, and there's a variation, and these 2 are the same. So for person number 1, it's kind of a homozygous on the reference SNP, and these 2 become a variation. And you can see this person 2 is a heterozygous, and this person 3 is a homozygous alternative allele. So it's completely different from the reference genome. Okay, and these colors here are 2 different SNP markers, and those red person are the ones are heterozygous variants, and homozygous variants, and those brown people, they are kind of heterozygous, and the purple people are homozygous for the reference. So you can see that in the case group, meaning the people who have specific disease, or this is a control group. In the case group, there's a more variation happens, and then you can see this particular SNP may be related, associated to that particular disease. Okay? And the underlying hypothesis of genome-wide association study is common disease, common variants. Okay? So this is very, very important. And common variants represent in more than 1 to 5% of the population contribute to common disease. Okay? So you can see this figure, and I encourage you to read this paper. This is a review paper in Nature in 2009. And by the way, it's also in the OnCourse system. You can take a look at that. So X figure here shows the allele frequency. Okay, so you can see this is the log scale. This allele frequency is 0.001. This is 0.05. Okay, and up until here, this becomes a very common variance, and this is a very rare variance. Okay, and the Y here really corresponding to the effect size. Effect size meaning that there are some variants you may have it, not have it, but if some variants are very rare, but if you have it, and you got into trouble. And those variants are really having big effect size. And so currently, our capacity is really to identify the association for the SNPs that locates in this area. So it can be very common, but not too much effect size, or it's very large effect size, but kind of pretty rare. Okay, and this part is very simple to figure out. So those are a few examples of high effect common variants influencing common disease. And those are already been discovered, most likely. All right. So the GWAS really focused on by this part, and our sequencing can help us to push towards the rare variants part. Okay. And the GWAS generally do not capture rare variants because they are based on the arrays, and the arrays are, by definition, that it has to be common variants to be put on the array. And the study design of GWAS study is hundreds of thousands of single nucleotide SNPs are tested for association with disease in hundreds or thousands of persons. So you can see a lot of data to go through. I mean, to do this type of experiment is very expensive as well. So the general study design, although there's some more other types as well, but the general study design most people will do still the case control studies, okay? So you got a group of people who have that disease or a group of people who doesn't have that disease. And then you genotype both of these two groups, and then you see which variant occurred more often in the disease population versus common population. So those kind of case control type of study. The second type of study is a cohort type of study, which means that study subjects are selected to be representative of base population and are followed prospectively for the development of numerous conditions. So one example is you measure the blood sugar level and to see how the genetic effect can really change that. It's not really specific to certain disease, rather it's more phenotype specific. So those are really the design of cohort studies. Okay? All right. There's one review paper, also from the same person, and published in 2010, New England Journal of Medicine. And this is a very interesting paper that summarizes all the significant associations reported through March 2010. Okay? And think about that, how much money we already put into the GWAS. Okay? And these are a summary of all the results that has been published so far. It includes 800 SNPs included in 545 studies and focusing on 150 disease and traits. So this is a very nice resource, very nice information here. However, think about that, it's kind of disappointing. And those are the only ones we identified, given the dollar amount of money that already put into the GWAS studies. Okay. And there are many limitations of GWAS. All right, I have to present some limitations, otherwise why do I study next generation sequencing technology, right? So the major limitation is the lack of functional information. So disease trace associated SNPs are not necessarily the causal variants, okay? So the variants usually identified in these GWAS studies are the SNPs on the SNP chips. They are only the so-called tag SNPs, okay, that represents this local genome region. And it's not necessarily, if you see the association, that doesn't mean that particular SNP is causing the disease. So they are not really causal variants. And the tag SNP in the LD region, LD is a linkage disequilibrium. So you can think of that part of the DNA region that always go away together, okay? So a tag SNP, so for example, this one tag is representing this entire region. This is another tag, this is another tag, right? And if you see that this particular tag is associated to certain disease, that really doesn't mean this SNP is causing the disease. Rather, something in this region is causing the disease. It can be the real causal effect, it can be somewhere here, okay? And identification of causative variants would require further studies. For example, resequencing this entire region to identify which variants happen there and which one is potentially causing the disease. And the second limitation of GWAS is the statistical power issue. So statistical analysis entails an enormous number of association tests resulting in high potentials for false positive results. So what that means is when we say the GWAS study, you think of 1 million SNP arrays. There's 1 million markers will be identified. And the most simple way to think of that, you have to calculate 1 million associations, right? And for this SNP, whether it's related to disease, this one is related. So each one has a p-value. So after you do this multiple hypothesis correction, it's really hard to get really very. So if you identify something, it has to have very, very, very small p-values, like 10 power minus 8 or 9 level of p-values. Sometimes to get to that level of significance, you really need a much larger sample size. And we know this GWAS is not cheap. So sometimes it can be hard. Very large sample sizes are needed to reduce false positives and improve the reproducibility results. but I'm not saying that next generation sequencing can fix it. And likely it will make things worse because there's more variants will be identified, the novel ones will be identified. So if you correct the multiple hypothesis testing, it's even in the worst shape. Okay, one of the potential solutions for this, for the GWAS study is multi-stage design, including replication studies using independent samples and to limit the number of false positive results. So you can see that if there's a 3-stage study, if there's about 5 variants in that, so basically they designed some, they made some benchmark data. So they pretend this is something associated to disease, and then they were trying to see whether they can find them out. And there are the first stage, so they can analyze 400 individuals, and this is the number of SNPs that they analyzed. And the power is not going to be very good, but they only focus on the significant regions, and then they further increase the size, but focus on much smaller number of SNPs, and then do a much larger sample size on an even smaller stage. And that will help you to really get into the much higher statistical powers. So this is the paper if you're interested in, and just goes to this JAMA publication. Okay? And so we already talked about 2 limitations of GWAS. The first one is the markers you identified are not really causal variations. And the second one is the statistical issue. Okay. And there's another very important paper, and actually the same paper we just mentioned. The title, I think, of the paper is called Missing Heritability. Okay. So the GWAS, this is also another GWAS limitation. So identified variants in GWAS typically account for only a small proportion of the heritability and disease risk. So even for some disease, you identify some genomic regions, but those regions can only explain a small proportion of the disease risk. But why that happened? Okay, and the median odds ratio per copy of risk allele for the published GWAS study is it's 1.33. Okay, and the potential source of missing heritability is why we haven't seen those heritability in the GWAS study. It's SNPs with prevalence less than 5%, because most of the GWAS study is common variants, common disease. So you focus on those common variants, but a lot of diseases are related to the rare variants, so you don't really see them in the GWAS study. And low frequency allele frequencies, which from 0.5% to 5%, are really rare variants, less than 0.5%. And those are not on the SNP array, so you're not going to see them. And the Southern Genome Project tells us there's more than 11 million novel SNPs in our human genome, which any one of them can contribute to the heritability of certain disease. And the second issue, the potential thing that of the missing heritability is the interactions between genes or between genes and the environmental factors. So what that means is when we do the GWAS study, so most people will look for the association between individual SNP marker with the disease phenotype, okay, and do it one by one. But there are certain cases like you need 2 things happens together to cause the disease. And for those type of things, it's really, really hard because you think about 1 million SNPs. If you think about every possible 2 pairs, that is a lot of a combination. How about 3 pairs, right? So those are the things that is really not solvable with the current computational structures. That's why the interactions between genes and the environmental factors are contributing to those, and they are not missing in the GWAS studies. Another one is contributions of structure variations, including copy number variations, inversion and translocations, and all these things. And the GWAS will miss it, but NGS, next generation sequencing, can really help. Okay. Any questions so far? Any GWAS guy are not happy? No? All right. Actually, those are the things that I just read papers that I've never done GWAS in my life. So if Tatiana is mad next time when she comes here, and let her to explain things. Right? Now I'm going to talk about the next generation sequencing enables breakthrough in genetics of complex disease. So keep in mind, I'm going to talk about two types of major application. The first one is in the research market, meaning how this next generation sequencing can really help us to understand the mechanism of disease. So this is the etiology part, okay? And there's another chunk, which is personal genome sequencing. It's also sequence the genome. And that is the second part of this topic. In this etiology part, I'm going to talk about 3 examples. The first one is, and all these 3 examples are published in Nature Genetics. And the reason I pick those are not that they are great or they are in the Nature Genetics. That's part of the reason they are in the Nature Genetics. But they are really representing 3 ways of using this technology to understand the disease. The first one is whole genome sequencing following GWAS, and this is for a disease called SSS. I don't know what that is. Sick Sinners syndrome. Okay, and the second example is, so the first one is the whole genome sequencing. The second example is exome sequencing. So you only sequence the exome part. And one results they got is study the Miller syndrome. And the third one is pooled sequencing and to study human complex 1 disorder. Okay, so let's go to the first one, this disease called SSS. This is published in Nature Genetics in this year, okay? And it's a wide range of electrophysiology abnormalities, including failure in the senescence node, and so pretty serious when you read it, right? Although I don't know what that is. Can happen at any age, but primarily a disease of elderly and often secondary to the cardiac disorders for the younger individuals. And the treatment is permanent cardiac pacing. Okay, so. All right, so they were trying to see what gene is causing this disease or contributing to this disease. And the step 1 they did is the GWAS study. Okay, they did a case control design, and the population is Atlantic population. It's very restricted population. Okay, and they have the case group. They have almost 800 patients and 37,000 controls. So they really put a lot of money into this study, it seems. And they use the Affymetric SNP arrays, I think, or Illumina, I don't remember which one it is, but with 1 million or 2 million SNP markers on the array. And when they do the association study, they use 7.2 million SNPs on the SNP arrays and based on the imputation. Okay. I hope Tatiana will talk about the imputation next week. If she doesn't, I will talk about a little bit, but I do need to learn it. So the point is, for this SNP arrays, you only find out the tag SNPs, and then you can get a more higher resolution and by some computational approach. So for the SNP arrays, you got 2 million SNPs, and you do the imputation, and you got 7.2 million SNPs. And they found the association between this disease and this particular SNP marker in the chromosome 14 region. That's the conclusion of their GWAS study, which is pretty nice. Okay, and but the problem is they find out this variant, this SNP, but it's tag SNP. It's not really causal, and go for the region marked by this region. So how can we know that which exact variation is causing this particular disease, or have stronger association? So their strategy is that they did whole genome sequencing on 87 individuals with average 10x coverage. So one genomic location, they covered twice. It's not very deep, right? Most of the whole genome sequencing, we want to have about 30x coverage or beyond to be able to confidently call there's a variation there. But this is the experiment they did. So 87 individuals, including 80 controls, and 7 SS patients, and 4 of them carrying this SNP they identified, and 3 of them doesn't have this mark. And the results summary is they got, after this whole genome sequencing, they got 11 million SNPs were called. That's a lot of SNPs, right? And there's no significant SNP out of 14.11q region. And again, this is the region they initially identified by the GWAS study. Okay, so there's no significant SNPs outside of this region was observed. Okay, it's kind of wasted, right? So I guess if they know it, they really don't have to do the whole genome sequencing. They can really just design the target array to capture this particular region and then do the further sequencing of it. That likely can be on more individuals. Okay, but this was what they did. But within this 14.11q region, the strongest association happens here. Okay, this particular variant, and it occurred in all 4 carrier individuals. All these 4 have it, and all the 3 non-carrier cases, all the 3 doesn't have it, and 1 carrier controls. 1 people that have this variant but is not having the disease also don't have it. So this is the strongest association what they discovered, and this is this particular variant is non-synonymous mutation in exon 18 of this MALH6 gene. It's a gene encoding alpha heavy chain subunit of cardiomyosin. Okay, and also they found there's no other significant association after accounting for the effect of this particular variant, and it seems this is a pretty good target to do the further study. Okay, so just think, not really trying to study their results, but think of the way they are thinking. They did GWAS, identified a region, did whole genome sequencing on much smaller samples, and then identified a variation. Looks like it's have much stronger association with the disease. It's not satisfying, not there yet. So what they do is they further did many studies to validate this rare variance. So they see this variant showed significant association in 87 samples, but are they representative? So the question here is, this is a good association in this 87 samples. 87 is such a small sample. How about other people, right? There's so many different peoples there. And what they did is they zoom in the region, which is exon 18 region, in many more individuals. And in many individuals, including 351 Icelanders and the same population, some of with disease, others are controls. They further did Sanger sequencing on exon 18 of all these individuals. Okay, and also they did another SNP array on the same individuals. So basically, they want to test how good this genotyping platform works well. And then they further did this SNP array and on another 500 individuals. So they didn't do Sanger sequencing, and which include 50 patients and 470 controls. So this is the data they have at this stage. They have SNPs on chip, genotyped, and this together is 900 individuals. Is my math correct? Doesn't seem so, but okay, this is considered as 900 individuals. And some of these SNPs on the chips, they were genotyped, and this region is a part of also genotyped. Okay, and then keep in mind that they have also this 37,000 individuals that were initially already genotyped. Okay, based on the chips. And then they further did an imputation in this local region and trying to see whether this, what is the result for this particular marker likely in this broader individuals. And then they look at association between these two, whether this case and control or this SNP is really something that's highly associated. And the results are great. This particular allele, the allele frequency is only 0.38%, okay? And they found the effect size of this particular one is 12, and it's really, really high. And the odds ratio is really, really high. And more strikingly, and this is a very interesting figure, so you can see X here are the year of birth. So these are very old individuals. This comes to younger individuals, okay? And this is the percentage of people who carry this particular variant and eventually develop into this disease. For the very old people here, 1920s or even older, and more than 50% of people who carry this variant that eventually develop into this SSS, this disease. And of course, with the younger population, they haven't showed up. They just need to wait, right? So this is one of the examples to see that how people can publish a paper in Nature Genetics, how much work they really need to do. So what we have learned from this study, certainly, yes? This particular one, I don't know. They didn't mention that. I haven't checked that. I haven't think that. All right. So what we have learned from this study is certainly not which gene is important, which variant is important, but the way of their thinking. So combining SNP genotyping and whole genome sequencing can facilitate the discovery of disease associated to uncommon variants with substantial effects. So really remember that figure, okay? And it's really pushed towards the left end of the figure with the rare variants, large effect size. And using high-throughput sequencing technology, this design extends gene discovery beyond the common variants, common disease hypothesis, which is used by the GWAS. And genotype imputation can be a powerful tool to improving the statistical powers for association studies, okay? And this study did not further test whether the identified variant is mechanistic. So whether this is a causal variance, we still don't know about it, but likely it is. Okay? So any questions regarding this to this example? Just think through the way they are doing experiment. Yes?
Yeah, so did they also follow up on the tag SNP and see how the tag SNP varies among the...
They did see. I mean, the tag SNP were in the initial genotype of the population, and they see the association, but not that strong. And then when they look at it, when they go back to this figure, and when they further some of them are based on imputation, others are based on the real genotyping, and they see this is much higher effects. So they did went back to see the tag SNPs. I'm not sure they do have the family structures in this study. This is a typical GWAS study, not really family-based study, but at least for this particular example. Okay? So let's move on to the next example, which is the exome sequencing, and they study Miller syndrome. As you can see, it's amazing that how little resource they use to publish this. And the Miller syndrome, I guess they are just getting lucky, and this is something that they can really identify from the from the exome sequencing. Miller syndrome is a Mendelian disorder and rare multiple malfunction disorders. So there's something to learn from this disease as well. And hypothesized to be an autosome recessive disease, but likely to be an autosome dominant disease. Okay? So what is recessive, what is dominant? I want to just briefly introduce it. And if this is the gene encoded for the color of the eye, okay? If this is a mother gene, is blue, and father gene is brown, okay? And likely, and the eye brown, so this particular individual, the color of their eye is brown, okay? And because the blue is a recessive, so you need both to be blue to have a blue color eye, okay? If one of them is brown, and you will have a brown eye, and that is the definition of dominant, okay? And why we do exome sequencing? That's another question. Why is that anything good with it, okay? But the reason, I think one of the major reasons is whole genome sequencing is still too expensive. We cannot afford that. So we have to do something likely to be more important. So positional cloning studies focused on protein coding sequences have, when adequately powered, proved highly successful of identification of variants underlying monogenic disease. And the clear majority of allele variants known to underlie Mendelian disorder disrupt protein coding sequences. Okay? That's the reason that they went ahead to do the exon sequencing, because they think the variations on the exons are more likely to be important, although it's not necessarily true. And splice acceptor and donor sites, and those are the variants that would disrupt the splicing mechanism and represent additional class of sequences that are enriched for highly functional variants and are therefore targeted as well here as well. So if some variation really mess up with the splicing sites, and those will be sequenced in the exon sequencing part. So it's already included in the data. A large fraction of rare non-synonymous variants in the human genome are predicted to be deleters, and this contrasts with non-coding sequences, and where variants are more likely to have neutral or weak effects on phenotypes, even within well-conserved non-coding sequences. So the point is, again, the variations in the coding region are more likely to be phenotypically important. That's why when we have some limited resource, and exon sequencing is still a good choice. Okay? And the study design of this experiment is they only did 4 individuals. 4 individuals. So, from 3 families. A and B, they are siblings from the same family, and C and D, they are unrelated. That's all. That's all they did. Okay? And the experiment they did was a single-end 76 base pair arrays, and they focused on 26.6 megabase mappable targeted exon regions. Okay? So those are the size of the human exon, although we know that for the other platform that we focus on about 50 megabase region. But this particular study is a 26.6 megabase region. And they did a 40x coverage per individual, and that is pretty standard, I think. And they focus on non-synonymous variants and synonymous variants, and that is more disrupting the splicing, acceptor and donor sites, and insertion deletions. So these 3 types of variants are the ones they focus on. And in A and B, which are the individuals in the same family, and they identify at least a single variance in 4,600 genes and 2 or more variants in 2,800 genes. Okay? So that's the reason they use the 2 individuals. If you only focus on 1 individual, you will see much more variants, but they want to see that in this family, and they have at least 1 single variance in 4,600 genes and 2 or more in 2,800 genes. And that narrowed down the list of the genes a little bit, although not too much. Okay, so why this the different models will lead into different results? Okay, so we talk about this Miller syndrome that people think is a recessive disease, but it's likely to be dominant as well. So they want to see whether it's dominant or recessive, and one way to do it that it doesn't assume anything, right? So initially they focus on this is a dominant model. Why it's important? Okay, why dominant model is important? So each sibling A and B or B was required to have at least 1 new non-synonymous or splicing variants or insertion deletion variants in the same gene. Okay, if this is a dominant disease, this is 1 individual and 2 chromosomes, right? If there's a variant, if this 1 gene goes bad, 1 variant causes this gene goes bad, and if this is a dominant disease, and this person is going to be sick. Does that make sense? So if it's a dominant disease. Rather, if it's a recessive disease, you need both these 2 genes goes bad. So 1 variant happens in each of them. It doesn't have to be the same one. It doesn't have to be the same variants. But both 2 genes goes bad, and this person is sick. Okay? So this is a very powerful filtering. If you think about this is a dominant disease or not a recessive disease, it's very, very different. And when Tatiana gives the talk, I'm sure that he will show you those kind of family paradigms and which one has a disease, which one doesn't have it. And one of the major conclusions they got from those are whether this disease is likely to be dominant or recessive. And because the dominant one or recessive one, your way of analyzing the data will be different. Okay, I want to give you some prime on this now, and when Tatiana gives talk, I hope that you can get into a little bit further. Any question here? Okay. All right. Variants were detected in the same genes among A, B, C, or D. Doesn't have to be the same variants. Under dominant model, one variant per individual. Remember that? Not a two variant. One variant per individual. And they have 8 genes left. 8 genes happens in all the 4 individuals. Okay? And under recessive model, they have only 1 gene left, which is this particular gene. Okay, I'm not trying to read it. Okay, presumably important. All right, and the conclusion for this part I didn't read very thoroughly, but they said the recessive model was favored over dominant model for Miller syndrome based on the observation that each case was a compound heterozygote for new this mutation, and 5 or 6 mutations were predicted to be damaging in this particular gene. Okay? So, again, this is something I think first of all, this is a very good experiment, right? They did exon sequencing. This is a very rare disease, and they identified its variants, and I think this is the first time they bring in this dominant recessive model helping us to understand which variant is potentially causing the disease. But sometimes it's not that easy. And when Tatiana gives talk, and we know it's kind of pretty painful as well. For this one, they did a good job, but at the same time, they got lucky. All right. The third application, okay, are we okay now? So the first application was whole genome sequencing, follow GWAS. The second one is exon sequencing. And the third one, I'm going to talk about the pooled sequencing, when people are poor, like us. We don't have money, too much money, so we have to do the pooled sequencing. Okay? So for this project, it's also published in Nature Genetics. I keep messing up this. I don't remember, it's 2010 and 11. But anyways, they publish it. So this disease is a mitochondrial respiratory chain disease, and there's actually many different forms, and complex 1 deficiency, 1 in 5,000 live births represented in infancy and very early adulthood. And it can be Lyme syndrome, and it can be many different type of things. It's pretty serious. Seems a nasty disease, right? And many genes are involved, mitochondrial, both mitochondrial and nuclear genes. Many genes are involved in this disease. The knowledge we have right now is roughly 15 to 20% of isolated disease cases are due to the mutation in mitochondrial. Rest are caused by nuclear defects. Okay, this is the knowledge we have right now. And 25 genes has been identified by the candidate gene and sequencing and linkage analysis or homozygosity mapping. So, which means this is a complicated disease. There's a lot of different way you can go wrong. But the bottom line is we do have some knowledge about this. Okay, it's not going to be the whole genome sequencing. And we already know there's some important genes are related to this disease. We want to further characterize it. Okay? So that is, so the general strategy here is they had 103 cases, okay, and 100 focusing on 103 genes, okay. So I'm not going to go through this because all these things will be talked about in the later slides, okay. So the pool they have for this sequencing is they have 103 cases and 42 controls. And they put 20 to 21 individuals in one pool of DNA sequence. And they have 5 pools of cases and 2 pools of controls. All the controls are actually coming from the HapMap samples. Okay? And they did a PCR amplification on 103 genes. I mean, it happens to be 103 as well, so don't worry about that. And these 103 genes actually have 653 exons. The overall region they did a PCR target is 145 kb region. It's not very long, okay? So for the pooled sequencing project, you better start from some biological knowledge. You already know this part is important, okay? Now I'm going to pool so many individuals, 20 samples into one pool, and then I need to run PCR to target, to grab the regions that I'm interested in. And so the region that they did PCR is 145 kilobase region, and with almost 1,000 PCR amplicons. Okay? Any questions on this? So they have the pools and they have the PCR product and pull down certain regions. And the platform they did sequencing is Illumina sequencing, and the coverage here is for 90% of nuclear targets. Nuclear targets are not the mitochondrial ones. It's over 100x coverage. Okay? Is that pretty big? Yeah, it's pretty deep in terms of 100. But think of you have 20 individuals pulled into one sample, and 100 coverage is not very deep. Okay? And but the median coverage is 3,400, the X coverage, so 168 per individual, and that is a pretty deep coverage for most of the regions. Okay, this is their kind of their coverage, and the results here is they detected 600 high confidence variants in the case pool and 264 low confidence variants supported by at least 3 reads on each strand. All right, just again, think of their study design. They don't have money to sequence every individual. Rather, they pull 20 individual DNA into one pool, and then they use the PCR to target this specific region and they did sequencing. Yeah? Oh, this one? Well, so basically they found that for the 90% of nuclear targets, okay, nuclear genes, okay, and so some of the regions they have deeper coverage, others they don't have too much deeper coverage, okay, and the median coverage is like a 50 percentile. We have 50% of the regions that the coverage is higher than 3,400 X, right? But that's only for 50% of it. But 90% of them is higher than 100x coverage. So that's it's just a 90 percentile or 50 percentile issue. No, it's used for everything. I mean, even for the exon sequencing, and you will be able to, they do have the statistics on what is the average coverage, the median coverage, 90% of the coverage, what 90% of the regions, what the coverage is, or things like that. It's average. It's average, yeah. Usually it is. So for the exon sequencing, we are more happy with 80x coverage at least. In many cases, we have that deep. Okay? And then they, before they want to make a story, they want to see the accuracy of the way they identify it. And because they have 2 HapMap controls and 2 pools, including 42 individuals, and they can consider that they already know the variants in these HapMap samples because those are already sequenced in many other places. So they found that with larger than 100 reads coverage, 100x coverage, and the sensitivity for those HapMap individuals are 92%. Okay, which means that 92% of variants in this region in the HapMap samples, they were able to identify it. Okay, the specificity of this is 99%. Okay, it's pretty good. Although we all know that this really doesn't mean much. given the large number of negative positions, right? Negative positions meaning the positions that there's no variance. So that's why that sensitivity is an important measure here, but I don't like the specificity. For many of these studies, if people publish on ROC curves, it really doesn't mean much because the unbalance between the positive and negative numbers and precision recall curve is a much better measurement for that. But we can get into that in the later lectures. For the rare variance, however, and the sensitivity is 86% sensitivity for doubletons. Doubletons meaning 2 individuals have it, okay? And 66% for the singletons. So you can see that the more rare the variant is, the more difficult you will be able to identify it using this pooled sequencing strategy. That makes sense. I mean, but the statistics are important. For the minor allele frequency estimated from read counts correlated strongly with the expected frequency in HapMap pools. Meaning that we got this one pool, 20 individuals, we can estimate the minor allele frequency of that particular variation. And for the HapMap samples, because the variation we already know, and we also know their real allele frequency, and the estimation, the correlation is very, very high. So that means the sequencer, the general strategy really worked. So this is a kind of a validation on their accuracy part because of they use so many HapMap samples. And the variance prioritization is the next step, right? So you identified 600 variants or 200 variants with low confidence. Which one do we need to pick? Which one do we want to focus on, right? And they want to throw out the variants that are not likely to be very useful. The first way to do that is they throw out the variants in healthy individuals, okay? Healthy individual meaning HapMap samples. They already have those samples. So because of HapMap individuals, they are not sick, okay? So if there's a variance happens both in this case and also happens in the general populations, it's unlikely to be very important in this disease. And they also filter out synonymous variations, meaning those variants that will not cause amino acid sequence changes. And we just mentioned that it's okay to do it, and that's a common practice. But there are a lot of synonymous variations are important as well, right? But that's the way they do it. They did it. They also filter out non-coding variants. So meaning that the variants are not in the protein coding genes. They throw those away. They assume those are not important. And they also filter out the missense variants with low conservations, okay? So for the ones that change amino acid sequences, but it's not really evolutionarily conserved, they think it's unlikely or less likely those have important biological consequences. So they filter out these individuals, and then they still have 109 variants that is in these highly confident ones left, and those are more likely. So remember, initially we have 600 something, right? And they found 109 left in this group and 107 left in the second group, which is a lower confident variance. And then next step, this is very important in pooled sequencing. I still want to stress out the point that I'm not trying to ask you to understand how this gene works or things like that. I'm trying to ask you to think through the general study design they use. They put samples together, amplify certain regions, and did deep sequencing, identified variants, filter out the unimportant ones, and so you got about 100 or 200 variants are likely to be important. Now, they come back to these individuals, and then they want to further genotype the rare variants. Okay, the variants, the 100-something variants they identified. They want to further genotype it in the disease and control samples. The reason they want to do that is the first thing is it's necessary to validate. All right, you can always identify is that real, and you want to use other technology to prove that. More importantly, however, and for the pooled sequencing, you will not know that You only know there's a variance there. You don't know which individual of this variance come from that pool of 20 individuals. You only know it happens in these 20 individuals. You wouldn't know which individual. So only after you do the further genotyping, it allows you to assign the variance back into the individuals. And then those kind of association type of studies will further be possible. The results here is they validated 84% of high confidence variance and 11% of low confidence variance. Overall, they validated 151 likely deleterious variance and corresponding to 115 unique loci. And these are our other general statistics. And for the 60 individuals with this disease without a previous genetic diagnosis, they were able to assign it back to which gene went wrong. And they can help to conduct a genetic diagnosis. Okay, so that's like the previous slide is not very important. But the summary here is the pooled sequencing identified variance in disease cohort and then further genotype on variance. Great approach using limited resource. You don't have too much money to spend. And that's why we have to do this pooled sequencing and only target on small part of the genome. Either save dollar or use the funds to genotyping more individuals. That can increase your statistical power. But pooled sequencing is not really perfect. There's a lot of problems associated with that. The first one is what if the causal variance resides in non-targeted genes? So you have to target on this 140-something kb region. What if the variance happens on other regions? So it really relies on a lot of your previous biological knowledge. And it can also on the non-target regions, but such as regulatory regions of unannotated exon, it was not detected. It can cause were not detected because of the lack of sensitivity, because you're pooling 20 samples into one pool. So it's much harder for the sequencers to figure out whether there's a variance or not. So the sensitivity can drop, especially for the mitochondrial DNA for their purpose. And if the variance contains full exon or gene deletions, okay, in one of the pools, if in one pool there's 5 people that have one gene deleted or one exon deleted, and which this approach you cannot really identify those. And were presented in the discovery screen but filtered out by the stringent criteria. It's possible as well. So those are potentially cause the false negatives. So these are something that really keep in mind. Again, for the next generation sequencing and disease discovery, so I talk about 3 examples, and there are really 3 types of experimental designs: the whole genome sequencing, follow the GWAS, the exon sequencing, and pooled sequencing. They were all managed, published in Nature Genetics, and they were really help us identify the causal effects or some of the associations. So we will take another 5 minutes break, and we'll come back in 5:10, about 10.